Good afternoon, everybody. Let's start our afternoon session that has a pretty amazing program, just like the morning was. And at first, I would like to call Szabó Gyöngyi Szabó from Harvard University, who is a physician scientist and an internationally known expert of liver diseases. Her laboratory investigates the complex role of chronic inflammation in the progression of liver diseases, studying the cellular and molecular mechanisms of inflammation. Her talk is about the power of translational research in liver diseases, what science accomplished in the past 30 years. Please.
Thank you. Good afternoon, Professor Mezey, the fellow academicians and Nobel laureates and friends and colleagues, ladies and gentlemen. It is a tremendous pleasure and honor to be here today at this 200-year anniversary of the Hungarian Academy of Sciences. I'm personally really humbled to be able to talk about this area of medicine and research that I've been having a privilege to participate in the last 30 years. So the liver has been recognized in the ancient Greek mythology. So the story goes that Prometheus, son of Zeus, was ordered to create a new creature, and then essentially looking at the gods, he created mankind that looked very similar to how the gods looked in those days. The problem was that these humans were very naked, and in a cold night they were very cold. So Prometheus took pity on them and stole fire from the gods to keep the humans warm. Now, the fire was the privilege of the gods only, and Zeus, learning about this, became incredibly angry at Prometheus and wanted to punish him. As a punishment, ordered that Prometheus would be chained to the Cascas mountain, and every day an eagle would come down to pick on the liver of Prometheus. But the liver regenerated, and that went on and on and on, and this was the eternal kind of punishment. What this highlights is that the special role of the liver, and in fact, even until now, we know that the liver has the special kind of feature of regeneration, and this is harnessed now for liver transplantation when, for example, in living donor donations, 2/3 of a normal liver can be removed from an individual to save lives of those who have failing livers. And the liver in both of the donor and recipient regenerates in about 6 weeks. Now, this story would be very simple, and if that remained, then I wouldn't be here talking to you today. Because what happens is that the normal liver in chronic liver diseases become fibrotic and cirrhotic, but this can go very silently. And in fact, we can lose 90% function of the liver without really having much of a clinical signs. So today I'm going to talk about what we have been interested in, and many others in the field of liver, which is inflammation in chronic liver diseases that is believed to be a driver of the progression of liver diseases from normal to fibrotic liver. And particularly, I will highlight some milestones that we kind of achieved in this field on hepatitis C virus infection, alcohol-associated liver disease, and metabolic dysfunction-associated liver diseases. So as a clinician, we always have to think about the big picture, but at the end, it comes down what happens between the physician and the patient. So I'm going to share this story because this was one of the patients who came to me early on in my independent kind of attending career. The 67-year-old, very nice gentleman who was a PhD scientist, a chemist, who came from Hungary, and then we had that special kind of connection speaking Hungarian in the clinic. But he was diagnosed a few years prior with hepatitis C, and in fact, he already had evidence of scarring, cirrhosis, but he was well controlled. In the winter time, they went to Florida to be there with their family, and then when he came back in the spring, he told me that in fact he had a decompensation event, but that was stabilized. So a few weeks later, he became ill and presented to the emergency room with fever and fluid in his belly and abdominal pain. He was admitted to the hospital where they took a sample of the fluid and diagnosed bacterial infection. Now, as a cirrhotic patient, infection is a very kind of bad outcome that can happen in these individuals, and in spite of treatments with antibiotics, he developed sepsis, multi-organ failure, and essentially he has deceased. So this is a case where acute and chronic liver failure triggered by an infection occurred in a setting of cirrhosis caused by untreated hepatitis C infection. The year was 2007. This should not happen today, at least not with this very fast course, because now we have treatment for hepatitis C. So how did we get there? So in 1999, the global epidemiology of hep C infection was about 180 million people were infected, and the risk of liver cirrhosis and cancer was very high in this patient population. In addition, about 3 to 4 million newly infected individuals occurred annually, and obviously this was a major medical issue and around the entire world. Furthermore, we really didn't have treatments, and patients with hep C over a period of 20 to 30 years or sometimes faster developed this decompensated cirrhosis and also hepatocellular cancer that was frequent in the setting of hep CV. And the outcome was either liver transplantation that was very scarce or essentially no treatment for these patients. And it wasn't until 1989 that the hepatitis C virus structure was discovered, and discovered that it's a single-serine RNA virus that has various genotypes and replicates only in hepatocytes. And the virus is encoded in a way that it has structural proteins and the non-structural proteins that are pivotal for the viral replication. And in fact, the protease and the RNA polymerase component are the ones that eventually were targeted for treatments that can stop the virus replication. But in the early days, what we know is that acute hepatitis C was very silent. A lot of people didn't even necessarily recognize that they have infection, but only about 30% of individuals actually cleared the virus. And the majority remained in this chronic hep C infection category where the virus continued. Even though people developed antibodies, that was enough to detect that they have been exposed to the virus, but the antibody was not protective. And the reason for that is because the immune system was not really ready to induce a robust immune response to eliminate the hep C infection. So we were very much interested in what could be the reason for this sort of low immune response against hep CV. So it turns out that we learned that the component of the virus, the NS3-4A protein, actually could undermine the host immune response, because normally these intracellular receptors could recognize the double-stranded version of this virus when it starts to replicate. But both the RIG-I and the TLR3 adapter molecules were cleaved by the hep CV virus. So very low interferon response happened, and essentially that gave rise to the chronic infection. In addition, we knew that inflammation was a major component in the progression of the disease, so we were interested in how is hep CV inducing inflammation, and we essentially discovered that some of the core protein and the NS3 actually could activate some of the surface receptors on immune cells, toll-like receptor 2, to induce pro-inflammatory cytokines that contribute to disease progression. And other studies related to some of the dendritic cells that are recognizing viruses and induce T-cell responses were also attenuated by hep CV. We showed all the plasmacytoid dendritic cells that produce interferon, as well as the myeloid dendritic cell type 2 that produce another type of interferon, interferon lambda, are subverted by hepatitis C infection, contributing to the impaired hep CV specific T-cell responses and elimination. So fast forward to 1999, then comes SARS-CoV infection, and the COVID-19 is also a single-celled RNA virus, although it's in a different family than hepatitis C. But it also affects the liver, and the SARS-CoV infected many other cell types. But we essentially use new technologies, and here I'm showing the single-cell nucleus and spatial transcriptomic analysis that we did with a group of people from MIT and in our institution at Harvard to essentially create an atlas of the human liver cells that were infected and modified by COVID-19, and I'm not going to have time to go into the details, just wanted to show that these new technologies will help us to further and better understand diseases in the liver. The success story of hepatitis C, however, remains. And I think this is probably the best and the most fundamental and impressive story, I think, in hepatology, at least in my lifetime, that since 1989, when the virus was discovered, we spent the first 10 years playing with all kinds of clinical trials with interferon that was very toxic to the patients, not tolerated, and really didn't clear the virus. But then once the replication in vitro system was established and animal model was established, that really opened the field and really facilitated the discovery of the various serine protease inhibitors, RNA polymerase inhibitors that now provide a 99 to 100% clearance for the virus with a 6 to 8 weeks treatment in newly infected or even chronically infected individuals. And obviously the story and then the impact of this was recognized by Nobel in 2020 when some of my colleagues, Harvey Alter, Michael Houghton, and Charlie Rice received the Nobel for this. Now, what we also learned and knew in the hepatitis C infection that some people actually developed the progression much faster than others. And one of the clear factors that contributed to this was alcohol use. So those who had alcohol and hepatitis C had a much steeper curve in developing progressive liver disease. So that kind of led us to the question that why is this important? And in fact, alcohol use has been increasing tremendously around the entire world in the past 10 plus years. It is particularly concerning that this is increasing, at least in the United States, in the younger population and also in women who are, by the way, more susceptible to alcohol-induced liver disease. So cirrhotic death due to alcoholic liver disease is significantly increased compared to previous years. And on the right lower side, if you look at the causes of cirrhosis, now the yellow one is alcoholic liver disease, and as you see, the green was hepatitis C. As we go into the current years, the curves go in a way that alcohol is the primary reason for progressive liver disease. So alcoholic liver disease can go very silently, but there is this entity of alcoholic hepatitis that is an acute event, and it could be a very deadly event for patients because about 30%, there's only about 30% survival in those patients. So what happens is that the probability of survival with the more severe disease is really exponentially decreasing because the mortality is increasing. But the really concerning thing is that the therapy that we use, corticosteroids, was established in 1971, and since then there is no new therapy. So this was something that many of us, and I think I was playing a major role in kind of bringing attention to this and trying to understand what can be done for these patients. So obviously we wanted to understand what is the mechanism of the disease, and it turns out alcohol affects multiple organs, including the liver, the intestine on the right here, and then the brain, of course, and I'm not going to talk about the brain, but now we know that some of the inflammatory processes induced by alcohol actually can contribute to the addictive behavior that comes with alcohol. So in the gut, which is on the right side, this pointer doesn't work very well for me. So on the right side, there is the gut, and in the gut, the alcohol has multiple effects. It changes the microbiome composition, it increases permeability, and essentially makes the gut leaky. But that allows microbial components to get into the liver very fast through the portal vein. And in the liver, these damage-associated and pathogen-associated molecules then turn on inflammation in the various inflammatory cells that are in the liver, resident macrophages, and so forth. Furthermore, they recruit newer macrophages and neutrophils from the bone marrow. So at the same time, alcohol can directly damage hepatocytes, that hepatocytes release various damage-associated molecules. So if we look in a patient with alcoholic hepatitis, we find in the red dots endotoxin levels. This is bacterial LPS, that component of gram-negative bacteria in the circulation. 16 is the bacterial DNA increased, and the host response is represented by the soluble CD14. And in fact, we find that some of these actually can be prognostic markers in these patients. But I wanted to go back to these danger signals because I think the discovery of this kind of came along with our studies as the field sort of learned more about inflammation. And turns out that there are pattern recognition receptors that are 90+% conserved between Drosophila and human to recognize danger signals for the immune system. endogenous danger signals that could be from damaged cells, or exogenous danger signals that come from pathogens, and it really allows discrimination between self and non-self. And on the right-hand side, I'm showing that there are about actually 11 different toll-like receptors that are specialized in recognition of various danger signals, but as you see that the downstream effects are the same, all go to inflammation in a very similar way. So this was, again, another major discovery that was recognized by the Nobel, and Bruce Beutler, Jules Hoffman, and Ralph Steinman received this for the discovery of the pathorecognition receptors and dendritic cell functions. So these toll-like receptors and inflammasomes play a major role, and in fact, this internal receptor system called inflammasome is a major regulator, secondary step regulation in inflammation that, when it's activated, can cleave interleukin-1, which is one of the inflammatory cytokines. But turns out that we also found that this can actually get out of the cells and be active in the circulation or being taken up by a cell that doesn't have this receptor complex. And this was particularly interesting in case of acute liver diseases, because we know that patients with alcoholic liver disease stop drinking alcohol, yet they have inflammation going on for several months. And they have no idea what maintains this inflammation. So we found that potentially these aspects, as induced by alcohol, actually could be a kind of reason how they contain and maintain inflammation. And in fact, we find that if we block the sensor molecule, the NLRP3, with this small molecule MCC950, at least in an animal model, we can attenuate and essentially stop this progression of inflammation. Another thing that we found related to alcoholic liver disease, that as alcohol or metabolic stress damages hepatocytes, these hepatocytes release danger-associated molecules, again, activate the NLRP3 inflammasome to produce interleukin-1 beta. And that interleukin-1 beta quickly binds to receptors on other cells, including hepatocytes. But what happens is that it has multiple effects on the hepatocytes, upregulating cytokines, sensitizes to damage, impair regeneration, and promote fibrosis. So we, in an animal model, asked the question whether blocking the IL-1 receptor could have a beneficial effect on alcoholic liver disease. And in an animal model, we got a beautiful inhibition of alcohol-induced liver damage, everything. Well, this triggered a clinical trial that was supported by the NIH and with 4 other centers. We had a multi-center trial where we essentially used a combination of this IL-1 receptor antagonist and anakinra, pentoxifylline that previously was shown to protect the liver, and zinc that was shown to reduce permeability of the gut, and had this clinical trial where we compared this combination with the standard of care methylprednisolone. And after, it was not fully enrolled, but essentially what we found was there was some signal, but there was no significant improvement in this IL-1 receptor antagonist group, although there was a signal for long-term benefit in the survival. And this triggered some other clinical trials that some of them are still ongoing. So in the alcoholic liver disease, there is another major component that we don't really understand, what is the role of neutrophils. So these are one of the innate immune cells that very rapidly are recruited to any kind of site of damage and inflammation. And we discovered that in liver disease, there is this kind of heterogeneity of these neutrophils, and particularly in alcoholic liver disease, they find different kinds of neutrophils, and transcriptomic analysis revealed that their profile is very different. There are some that are highly inflammatory, whereas some others that are very much exhausted. And my graduate student very nicely showed that the low-density neutrophils essentially are formed by the way that the nucleus of this normal high-density nucleus neutrophil is spilled out, and this is a form of extracellular traps that results in the surviving low-density neutrophils that we believe contribute to increased infection in these patients and also activation of immune cells. So these neutrophil extracellular traps are composed of multiple components, including citrullinated histone, extracellular DNA, neutrophil elastase, and so forth. And what we discovered is that this is a way of the neutrophils to actually activate other cells and have kind of crosstalk between cells, because the neutrophil extracellular traps, even in the absence of the neutrophils, when the cells go away, they still can activate macrophages to become inflammatory, and most importantly, activate liver stellate cells that are producing collagen for the maintenance of fibrosis. Another way that we can trying to understand how liver works is related to microRNAs. And microRNAs are small 2022 nucleotide sequence non-coding RNAs that can have effect on microRNA target cleavage, transcriptional repression, or various deacetylation processes. So in the liver, it has been recognized that various cells express various microRNAs, and in fact, these have roles in the liver disease, particularly in steatohepatitis. And one of those microRNAs is microRNA 122 that is predominant in hepatocytes. But turns out that in case of hepatocyte damage, this microRNA 122 spills out from the cells, but even within the cells has a multiple roles on increasing inflammation and fibrosis. These microRNAs coming out from the cell in the circulation can also serve as biomarkers, and that's an area of discovery. Another microRNA that is receiving attention in liver disease is inflammation-related microRNA, microRNA 155. And recently it was shown that it is involved not only in steatotic liver disease, but also in liver fibrosis. And in this particular study, we showed that if we used an inhibitor of the microRNA 155 and delivered it with an AAV-mediated vector, we could actually attenuate or even prevent fibrosis. So these are all areas that we are exploring and the field is exploring for the future. And I couldn't help myself to point out the microRNAs for this year, because Victor Ambrose was my colleague at UMass, and actually we worked on some of the microRNA trafficking experiments with him that is highly recognized field. So the interesting thing is that these microRNAs, although they are fairly stable in the circulations, many of them actually are encapsulated in these small extracellular vesicles, and particularly exosomes that are about 60 to 100 nanometer size extracellular vesicles coated with a double layer of plasma membrane actually are produced by the cells in an active way. And it turns out that these extracellular vesicles contain various components, particularly microRNAs that could be targets for biomarkers. What we also discovered is that alcohol, for example, or even high fat, induces production of these extracellular vesicles, particularly exosomes from hepatocytes, and here I'm showing the electron microscopy. And then these exosomes are loaded with microRNA 122, which is the hepatocyte-specific microRNA. Interestingly, these extracellular vesicles can be taken up by other cells, so for example, inflammatory cells, and these extracellular vesicles then reprogram the recipient cell to be more inflammation-oriented, or in case of macrophage-derived exosomes, to be more quiet macrophage in the future. So while we were working on these alcohol-associated liver diseases and many of those mechanisms that I showed you, it was discovered that we have another epidemic and pandemic on us from the standpoint of liver diseases, which is the metabolic dysfunction-associated steatotic liver disease. This is a very long name for a disease that essentially is the liver manifestation of the metabolic syndrome and obesity and type 2 diabetes. And this is increasingly common in patients with obesity, metabolic disorder, or cardiovascular disease. And if you look at these maps, it shows that between 1994 and 2015, the prevalence of obesity on the left and diabetes on the right is just tremendously increased in the United States. And for many years, it was kind of thought that this is a disease of the United States because McDonald's and all the bad kind of fast food. But turns out that the progression of this metabolic associated liver disease actually is happening around the entire globe. So now if we look at the WHO data from 2022, it shows that not only the US but other parts of the world are involved, and the global prevalence of the type 2 diabetes is also tracking all of this. And all of this leads to a high burden of the metabolic-associated liver disease around the world. So it's estimated that about 1.3 billion people worldwide have this disease, and the global prevalence is estimated at 30%, which is pretty astonishing. So the mechanisms how the metabolic dysfunction associated liver disease works are very similar to alcohol, but it's a little bit slower, to a slower extent. And of course, lipid metabolism and dysfunctional lipid metabolism and adipose tissue contributes to this too. But I just wanted to show that as opposed to the few clinical trials that I mentioned in alcoholic liver disease, in metabolic-associated studies of liver disease, actually there are about 120 or so clinical trials that were ongoing last year, and we have one drug that is approved so far, resmetirom, but all of these other targets are under investigation. Most recently, and this is hot off the press, New England Journal published this paper, which is a partial readout of a phase 3 clinical trial using the GLP drug semaglutide in this metabolic-associated steatohepatitis, where they found that individuals who received the semaglutide, there was a higher resolution of the previous fibrosis and also reduction in liver fibrosis. So this is an ongoing study, but it's very encouraging to see this. So the new nomenclature, what is coming for the future? Well, one of the excitement in the hepatology field, and I'm not sure how exciting is that for those who are outside of hepatology, but it kind of makes sense that after I showed you the huge prevalence, the 30% of the people in the world essentially being potentially affected by the metabolic liver disease, but let's be realistic, many of those people probably drink at the same time, right? Question is how much. So we as hepatologists now recognize that yes, of course, these two can go together, but we really don't know how is this going to have any effect on the progression of disease. So there's this new disease phenomenon or clinical nomenclature of the MET-ALD, which is a metabolic and alcoholic liver disease together, that we are studying, and essentially consensus meeting actually just tomorrow is happening between the American and the European liver societies to come up with some strategies how to go about this and how to plan clinical trials for the future. Looking ahead even more, I didn't talk about hepatitis B infection, and chronic Hep B is very prevalent, particularly on the eastern kind of part of the globe. And in Africa, 224 million people are affected, and Hep B is a precursor for hepatocellular cancer. So it's really encouraging that the WHO actually has the global elimination of viral hepatitis. And also, I think we have now new technologies and tools with molecular cellular therapies and so forth. And I hope that in the next iteration of the science, we can talk about new therapies that can help to restore the regenerative capacity of the liver. So with this, thank you, and I acknowledge all of those who contributed to this and NIH funding, of course. Thank you.
I would like to thank this beautiful presentation.
Thank you.
And I hope I didn't scare you. You still had 3 minutes left.
Oh my gosh, I think I went very fast. Thank you
Thank
so
you
much.
very much.
Thank you.
Next, I would like to ask Professor Eva Tardos from Cornell University, who is interested in algorithms and algorithmic game theory, the sub-area of theoretical computer science theory of designing systems and algorithms for selfish users. Her research focuses on networks and simple auctions, designing algorithms and games that provide provably close to optimal results. The title of her talk is Stability and Learning in Strategic Games. Please, thank you.
Thank you so much, and thank you for the opportunity to talk here. I guess I want to, before I'm talking about something technical, I want to thank the Hungarian science community for, getting me where I am. I was a timid undergraduate, even graduating from the maths major, which is what I did here. It didn't ever occur to me that I could be a scientist. And I guess I definitely owe a lot to András Frank, who was my advisor and who got me started on this journey, but actually the whole Hungarian community, like, Miki Shirovic and Lovas and many of them who encouraged me and convinced me that this is an option I could do, which totally didn't occur to me, to be honest. And I guess on top of it, maybe I want to especially dedicate this talk to Vera Sors, who I very warmly remember when this was the first event I ever attended in this room in the academy, when she became an academy member. And it was very memorable for me, and I'm sorry that she passed away. Okay, so I actually don't have anything as relevant for people's health to offer as the previous talk, so hopefully you still find it interesting. What I want to talk about is I'm currently in a computer science department. I still do mathematics, which is the section I'm representing here, but mathematics related to computer science. And I sort of changed from the kind of thing I used to do to something that involves game theory. And I want to start by explaining to you why I did that and what actually causes me to want to think about game theory, and what does it mean to think about game theory. So I guess the change, which kind of happened around 2000, at least certainly I became aware in maybe 99, 2000, is the agenda of how, when you want to think about algorithms or computing, how you think about it. Classically, and I guess I put it here before 2000, the computer takes care of tasks for us, like we want to schedule something or plan something or even plan the economy. You have the input or the data of what's happening, and then you design it, and that's what the algorithm takes care of. And what changed is that many things that we use algorithms to do are actually not standalone tasks. That is, you, as you log onto your computer, want the data to come to you. That's an optimization your computer takes care of you to try to get you the data you want. But it's the same network that the rest of them want to get data for themselves, and hence we interfere with each other, we're causing something for each other, and we're sharing the system. And as a result, we better design our algorithms and our systems so that when we individually optimize, we're not ruining each other's life. So I guess there is a classical danger, which goes under the catchy name of tragedy of the commons, that rational selfish action can ruin society. And I guess the example here is coming from an English village, and maybe I won't go through any sort of mathematical model, but the basic story is that the rule in the village was that anyone in the village could put a cows on the commons, hence the name tragedy of the commons. But the village grew and grew and grew. There were too many people there, and those poor cows were starving because it didn't look like this. It looked like too full with cows and not enough grass there. So everyone selfishly thought that putting my cow there is still better than not putting it there, but turns out at the end there wasn't enough milk for anyone. So that's the danger. Individually, people selfishly optimize, but something it goes wrong. This was called under the catchy name of price of energy, but basically the message is that what we want to understand is how do you design system or how do you design algorithms so that when every individual selfishly optimizes for themselves, which is what the computers do for us or what algorithms do for us, we don't ruin each other's life. Okay, my favorite example from the last many years since I worked on this was actually, you can decide whether it's car traffic or packet routing. It's a very simple model of routing traffic, which can be packets or cars, where if you think of cars, it's a very easy to realize that when there are too many cars on the road, it gets slower. So this is one way. When I choose my shortest path to get to whatever my destination is, then I'm a little bit ruining everyone else's life because I'm going to be one of those cars, and the rest of you experience me as a traffic. And too much traffic is not good for you. So that's sort of the simple model. This is a classical model, both used in computer science by the networking people, where they think of not cars but packets, and also think of the civil engineering community for modeling car traffic. Here is a simple example, just to have you, if you haven't seen it, I think this for the last 20 many years I work on this, I guess remains my simple example. It's the following super simple model of how, just to illustrate what can go on. There are 100 cars here that want to get from A to B, and there are two kinds of roads, one that takes an hour. Those are the black ones, and the red ones are the ones that are congestion sensitive, and there are X cars on it, then it takes X over 100 hours. So, 100 cars would take an hour. I guess if these 100 cars go 50-50, then 50 over 100, it's only half an hour. So this would get everyone to their destination in an hour and a half. People are annoyed. An hour and a half driving is annoying. So someone says, oh, I have a cool solution here. I put here a tunnel. You can just tunnel over, say, to make it extreme, in 0 minutes. You jump over there. And everyone says, oh, that's very cool, because now I can get my destination in an hour. All I do is, go from A upwards, and then down, and then across, and, X and Y were 50. Basically, I get across for an hour. Oops, everyone does that. And now suddenly the traffic went up to 100 each, and they're taking 2 hours. And what's worse, once the tunnel is there, you can't actually undo this, or at least it's super hard to undo it. We should just take out the tunnel, because individually, when people switch back to the old path, it's not actually helping. Now every path is taking 2 hours. Congestion made life worse. Congestion caused life to be worse. That's called Bray's paradox. Bray was a German scientist. I discovered this in the 60s. And I guess the paradox here is that an added option, there's an extra option there, made life worse. So this is actually a pretty mild demonstration of the tragedy of the commons. And I mean mild because it doesn't seem all that tragic. It went from 1.5 to 2 hours. So maybe it's not that bad. But certainly an illustration that selfish optimization can make life worse. Remember, if they only knew to stay away from that central edge, everyone would go home faster. So that's exactly the example that caused me to start working on this and trying to understand what's going on. And I guess our original paper with, I have the pictures of my co-authors here, with Tim Ravgarden, who was a student at the time and now a faculty at Columbia University. We proved that actually, at least for packet routing, life isn't so bad. And our theorem says that if you design the network to be able to carry twice as much traffic as what you're going to have, then you shouldn't worry about this. Life will actually be okay. So it gives you a way to counteract the trouble. It says that yes, selfish optimization by these packets, or simplistic optimization by the packets, can make life problematic, but I have a solution for you: make the roads bigger. Now, I understand in civil engineering this is not a good message. It's not that easy to make our roads bigger. But turns out in packet traffic, it is a pretty good message. And in fact, the networking people like it. And that's what they're doing, just putting down bigger cables, and that takes care of the problem. And they know this. So, okay, this was like 20 years ago. And what I'm hoping to tell you a little bit is something that maybe you're interested on in any way, that this is, oh, sorry, backwards is not working. This previous theorem made the simplistic assumption that the traffic, somehow everyone knows how to optimize how to drive, or even worse, the packets know how to optimize how they. Finding this sort of stable Nash equilibrium solution, which I showed you on a tiny, tiny graph, is not actually easy, and there are lots of problems with it. I guess maybe the red and the black one is the one I want to more focus on. First of all, for every packet to know what is optimal, they have to know about all the other packets, because it's a global optimization problem. Good luck. How do you know who is reading what? And second, because I'm currently a computer scientist, there is a technical computational difficulty. You actually computationally hard to find this equilibrium. So again, good luck. You can't find it. But here is something you can do instead, and here is literally what people are doing instead, is these are repeated interactions. Even driving is repeated interactions. Even if you, one time in your life, drive to Boston, or, drive to Bonn or someplace, you're using Google, and Google giving you directions. That's, for Google, it is a repeated interaction. They're repeatedly giving advice to people. If you think about packet traffic, that's routers are optimizing, and that's definitely repeated for them. And what these machines are doing is using a very simple learning algorithm. They're trying to learn what is the right thing to do. They don't take the global traffic, because how would they know? Instead, they're trying to give advice and hoping for the best, hoping that they learn the right advice to give. So here is, again, I'm representing the maths section. So here is a mathematical view of what I might mean by that they're learning to give the right advice. So, interaction happens over and over again. This is extra good for packet traffic, because one packet doesn't matter so much. Time goes on, and I'm hoping that they will satisfy this condition, which I call no regret. And I want to somehow maybe use, on the picture on the side here, how do I point to this? Oh, there. I want to use, this is my path, how I came here this morning. And I guess Google told me that there are many different ways to come, and I tried one of them. In fact, I didn't do what he suggested. I did one of those things. I guess I'm claiming if I do this over and over again, or if a machine does this over and over for us, if there is, it could be that, on days it rains, one should take the metro, and it rains that it's actually sunny, the tram is better, and I don't know, when the crowd is big, then buses are the best. I wouldn't know. That's very complicated. But there is a single solution that's consistently better than what I was doing. I didn't learn. I should have discovered that there is a good solution out there, or my algorithm didn't learn. So that is the condition. I guess the assumption is that whatever I did over time, my utility or the time I took, I should be better than any fixed solution with hindsight. If there is something that consistently was good, and I'm not doing as well as that one, I didn't learn. That is the assumption. I put in a little bit of an error bar here, which is a little bit of error over time t. Obviously, in the beginning, as I'm trying to learn, I'm going to make mistakes, so that's okay. But if the error is proportional to the time, the more time I spend, the bigger and bigger the error gets, then I somehow didn't learn. That is the assumption. And I want to think of this as a behavioral assumption. There are actually algorithms that guarantee this. But I want to think of it as any decent learning algorithm should satisfy this. And then I want to say, what can I prove? What can I say about the traffic if I make this assumption? I do have empirical work trying to look at certain, actually not in packet traffic, unfortunately, certain places where I do have that learning algorithms may or may not use our simple rules that guarantee this, but they do seem to satisfy the condition. So this is not crazy. I hope by telling you that it sounds reasonable to say that if they didn't satisfy this, they didn't learn. So here it is. And actually, in the early years of when my community started to think about this game theory, I guess the notion of price of energy was introduced by Koutsoupias and Papadimitriou in '88. And then, roughly 10 years later, a group of us, or a group of many people, proved that if everyone satisfies my no regret assumption, which I guess I now know how to go back to, then turns out the same proofs will work, and we even proved that you don't even have to the traffic can change over time and it's still okay. And generally the message was that no regret is both most natural and strong enough assumption, and decent learning will achieve this, and everything is as good as can be. And we can use this instead of the equilibrium assumption, which is, again, hard to find, both informationally and computationally. Okay, I want to tell you a little bit more of what I've been recently working on, and that, in fact, will be a criticism of this with some results. So one important assumption in each of these works is that as you iterate, if you remember my picture of time goes on in times, we have a clean copy of the game with no effect between the rounds. So my question is it reasonable? And I'm claiming some application it is, but many it's not. So here is back to my traffic examples. I claim in morning rush hour, it's very reasonable. No matter how bad morning traffic was on Monday morning, the cars are gone Tuesday morning. Like overnight, they went away. It depends on the scale of time, actually, not whether you cars or packets, despite my picture here. If I do millisecond by millisecond packet traffic, then it's absolutely not true. The packets that caused the trouble a millisecond ago, they're there. They're getting sent again. They're still causing trouble. Honestly, the difference is not between packets and cars. The same happens on the car side. If I change the time scale from not morning rush hour, but I call it, if 5 minutes ago there was a traffic jam at some place, then it's still there, I guess, likely. Maybe it went away, but very unlikely, so probably it's still there. So what happens if there is such a carryover effect? And what can we say about learners or reasonable assumptions on learners with the outcome in games where there is such an effect? And the game that I've been will tell you a little bit about is actually a packet-based game, very similar to online packet routing and joint work with my more recent student, Jason Gaetonda, where we're thinking about how packets work on the internet. So let me give you a sort of very high level and admittedly oversimplified version. So here on the blue side, there are some packets are arriving, and that's the traffic, like you requesting some web pages to see. They have to go to some servers, but servers are limited. They can take at most one packet at a time and serve it, and maybe they're not even perfect. Sometimes they fail at serving it. And the way the internet works, if you forget, if a server is unable to serve a packet, you're just going to have to resend it. That's the carryover effect. If you fail at a packet, it stays in the system. It wants to go back again. So we're trying to model this in a very simple mathematical model. Here it goes. Some queues where packets arrive at some rate lambda, and servers that can serve them at some rate mu. And I guess the rules are one packet at a time on both sides. And then unsuccessful packets get returned. And one thing that at least networking systems do nowadays to help this is packets have an H stamp in them, and they favor the old packets. Turns out this is very useful. That helps clear the system, that nothing stays around forever. So I guess to tell you what we could show here, first of all, one can somehow, maybe naturally you'll accept, you believe me, and this is actually very similar to conditions that we just heard Laci Lovász talk about in a bit simpler setting, is that if I could coordinate, if I do global coordination, then this is what would be the condition. For every subset of queues, so take these two of them together, there must be two servers that have enough capacity to serve them. Every subset of S queues should be a subset of servers not bigger in cardinality. Why not bigger in cardinality? Because I said sending it one place at a time. So, if I go back to the single queue, then, for this queue to succeed, even if he's alone, there has to be one place he can send it. He can only send one thing at a time. Similarly for two packets, two queues. That's the condition. It's a little harder to prove than the things that Laci didn't go into details of proving, so I won't go into details of proving either, but it's very analogous to Laci's condition. So the question is, if we're not centrally coordinating, what will happen? And here is the pair of theorems that I want to tell you about. One is we need a 50% increase in capacity, just like it was in the Nash equilibrium version. That is, if everyone just learning, then you need twice as much capacity, but twice as much will do it, which is interesting. It does say that it's, no regret learning is not that bad, but maybe surprisingly, if I actually get these guys to find a stable solution, I don't need twice as much, I need a little bit less. So it also illustrates that turns out in this setting, no regret is not perfect. You should be able to, or we should be able to do better. And I guess maybe give you two interesting things before concluding. One, I want to give you a tiny bit of a sense of why is it that this factor of 2 extra and the no regret condition go together and actually guarantee that things get across. I won't do a proper proof. I want to be accessible to a larger set of audience, but I want to give you a sense that it is that given this condition, the band makes sense and it will be tight. And in fact, it is tight. That is, less than 2 is not enough here. So why is this? We're using a theorem that maybe you believe from probability theory that says that if I have a numbers chopping up and down, that's a random process, just a bunch of numbers going up and down. They don't jump too much, which is what the word sufficiently regular represents, by which I mean they never go too much up and too much down in one. And when they're high, then they tend to come down. So this is a process that, sometimes goes up, but once it's above some height, it has a tendency of coming down, and you won't be too surprised to hear that this says that it will stay banded. In expectation, we use this in thinking about the sum of ages of all the packets. And here is sort of a one-slide idea of the proof. Focus on some age of packets, some older packets, and I guess say, okay, these guys have priority, so why don't I think of them separately? And there are 2 cases. Either these guys were smart enough, they found the right servers, they went there, they had priority, they got served, say half of the time. Okay, then we made some progress and we're okay. Or, I guess alternately, they were dumb, they didn't go to the right places. But the good servers were half of the time empty. They should not have regret. Wait, they could have done there. It was empty. They could have been served there. They had priority. So then they get served. This is why I need the factor of 2. Either they went there half the time, and that's good enough for me, or the thing was empty half the time, in which case the regret condition helps me. Hence, I really need the factor of 2, and then I do this for every age group and sum it, and don't worry about it. What I want to show you is the example of what's wrong with it, and that's sort of the open directions that we're currently working on. So here is a very simple example. Again, 2 queues, serving packets, service rates. Unfortunately, okay, first of all, what I illustrated, that both of them deterministically send to the better server, that's a no-regret solution. As you can see, there are 2 of them, they're sharing it 50-50, and the other server is apparently pretty bad, less than 50%, so that's not working. You also see, of course, that they're in trouble. There's linear growth here because the sum of the arrival rate is bigger than the service rate, so it's never going to work out for them. But what's painful is if one of them were to deviate, and looks like this is an altruistic mode, he's helping his buddy, who's 10% of the time will be alone, actually everyone gets through. It's not, it's selfishly better for them. Why? Because he first of all helped his buddy, so his buddy will run out of packets. But it's also the case that whenever he goes there, he will be the older packet, so he'll get priority. So he definitely will have regret. He thinks that up is better, but turns out it's not true. And that's because, and I guess I maybe skip those slides, our notion of no regret is a bit too myopic, and people should be sort of paying more long-term attention of what's going on in their life, which the example shows. But I'm going to maybe stop here with some conclusion. So I think understanding what happens in large systems when instead of we globally optimizing, every player selfishly optimizes, I think is an incredibly important agenda because this is how our life works. Certainly this is how our life works on the internet. This is how our life works in any system on the web, any place where we interact, where we have effects on each other, which is almost everywhere. It's interesting to what one can do. I guess with the example, hopefully I convince some of you that no-regret learning might be a bit too simplistic here, but still actually we can prove some positive things, and I guess the open agenda that I'm currently working on is what would be a better assumption and what are matching learning algorithms that are better. And I'm going to stop here, and thank you for your attention.
Thank you very much for
Thank
the
you.
excellent talk.
Thank you.
Please accept this.
Thank you.
MPA.
Thank you so much.
Thank you very much, and I would like to ask Professor May-Britt to give her talk. She received her education as a psychologist at the Department of Psychology, University of Oslo, and obtained her PhD in neurophysiology at the Faculty of Medicine. She shared half of the Nobel Prize in Physiology and Medicine in 2014, awarded for work concerning the grid cells in the entorhinal cortex and its contribution to memory formation in the hippocampus. She will talk to us about spatial maps in entorhinal cortex and their origin. Please.
Thank you so much for that introduction. And do you hear me at the back too? So happy birthday to the Academy. 200 years is not a bad year to celebrate, but thank you also for inviting Edward and me here to present some data from our lab. Since this is the Academy of Science, I wanted you to read the first sentence here, the power of science. I think that is the vision of the Academy, that through science and knowledge, we can move the world forward and not be selfish, hopefully. My field, neuroscience, we want to use the algorithms to uncover how the brain is working. So we can't study the whole brain, so we have to focus, and what we have decided to focus on in our lab is the GPS system, to say it in a common way. We want to understand spatial maps in the entorhinal cortex and their origin as a model system for the rest of the brain. I was so lucky that even in high school, I was with this man sitting here in the front row, Edward Moser. We became good friends. And through the 80s, 90s, and up till now, we have worked together and trying to understand the hippocampal formation. And as I said, we want to understand the higher cognitive functions and recipes that the brain is using to generating such functions, like our memories, ideas, and whatever, but now focus on space and memory. This is such a beautiful work by a Hungarian, Laszlo Szeres, do I say it correctly? And he worked so hard to dissect out this structure of the brain, which is called the hippocampus, and you see why it's called the hippocampus, because it's Latin for, or Greek, I think, for a seahorse. And why is this structure not only beautiful, it's so important? Because what we have learned, even since the 50s, that when patients having epileptic seizures and medicine couldn't help them, they had to remove the focus of the epileptic seizures, all the electrical currents in the brain, and they removed both of the hippocampi on both sides. And what happened then is that these patients, and one of the most famous of them, age M, could not remember any events, no episodes, could not make sense of time, what happened before other things, and could not find his way even from his room to the bathroom and back. So this made neuroscientists very curious about how is this hippocampus working? But how can we approach and to study this structure? What we know is that the brain is full of cells. In the human brain, we have more than 80 billion neurons, and they talk to each other by sending tiny electrical signals crossing the synapse with the help of chemistry. And John O'Keefe, that Edward and I received the Nobel Prize with in 2014 together with, he wanted to understand what is this hippocampus doing. And in order to do that, he placed tiny sensors close to the cells in the hippocampus of behaving rodents. And the rodents were motivated by chocolate or rice, crumbs or whatever, and he was throwing that out so that they were just walking freely in open space. And then he was listening to one single cell at a time, and he tried to understand why is this cell active and why is the other cell silent. And here we should hear some sound. Yes. So it's not a beautiful sound, but it's a sound of a hippocampus cell. And one red dot, that means one such electrical signal from this cell. And when you hear the pop, 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 that means that this cell is active. What you see here is that this cell is most active in the northwest corner, and it's almost silent in the rest of the box. And then if you had been John O'Keefe, you could think maybe there's a specific odor or something that happened in that corner. Why is this cell firing exactly in this position and silent in the rest? Of course, John, and this is just another way to show it's a rate map, and with warm colors where the cell is very active. So, of course, John recorded many cells, and others also. And then if you record many, many cells, you see that different cells, they have different preference for different places in this environment. And you can even use this activity to know exactly where the animal is with an error of only 5 centimeters if you record for some time. So John O'Keefe decided that these cells should be called place cells, and their function in the hippocampus and in the brain is to give the brain a position code. At almost the same time, but then published later, these two worked together, Jim Rank and Jeff Taubey. And they studied another structure that is close to the hippocampus, and that is the dorsal presubiculum in yellow. Maybe we can, yes, you see it here. So when they used the same technique as John, having tiny sensors in this structure, what did they find? They found different types of cells. These cells, they responded to the direction of the head of the animal. So if you record from me now, then one cell could be active when my head is pointing this way, and then it's pop, 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 silence, pop, 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 silence. And then again, different cells would be active towards different directions. So, no surprise, these cells, they provide a direction code to the spatial navigation system. And these guys decided to call these cells head direction cells. So Edward and I, we did our PhDs at the University of Oslo with Per Andersen as a mentor, and he gave us so many good advices. And one good advice was that if you want to learn something, go to the one who is the best in that field. So he encouraged us, and he also organized so that John O'Keefe welcomed Edward and me to London so that we could learn this technique to record from these brain cells. And we brought our 2 young daughters, and we had a really good time with John, and we were well trained when we moved to Trondheim in Norway. And when we started our own lab, half year after our PhDs, we decided that we wanted to understand where is the hippocampus getting all this spatial information from? Is it generated within the hippocampus itself, or is it coming outside? And how could we address that question? We had, of course, wonderful PhD students that we worked together with, and we decided that if you check the anatomy of the hippocampus, and the hippocampus is so beautiful also in the rodent. This is, in fact, a rabbit brain drawn by our supervisor, Per Andersen. It looks like a beautiful sausage. Now I'm almost a vegetarian, but still, hippocampus looks gorgeous. If you slice it like a salami, then you see a repeated pattern inside. And this repeated pattern of connections is going then from the entorhinal cortex into the dentate gyrus, into CA3, up to C1, back to subiculum, and then back to entorhinal cortex. So what we asked at this time was, if we block the activity either by tiny, tiny, tiny micro knife cuts in the fibers there, or ibotenic acid lesions, do we still see spatial signals in the hippocampus? And then we recorded from CA1 in the hippocampus, and simsalabim, there was a spatial signal. The cells still had the beautiful place cell activity. We knew at that time the anatomy, so that we knew that C1 probably didn't make this or generate this spatial signal because there's no recurrent connections between C1 cells. So we decided then to go upstream of the entorhinal cortex, the input structure, which was the entorhinal cortex, and we decided we put our sensors there. People had been there before and concluded that there were not much spatial information in these cells, but we worked together with May-Britt, and he suggested you should go into that part of the entorhinal cortex and check that part that is giving information to the dorsal hippocampus where John O'Keefe discovered the place cells. And this is the back of the rat brain, and this colorful area is then the entorhinal cortex. And we, as I said, we had our electrodes there. And what did we find? I have to explain this first to you. So this is the black traces. That is the trace of the animal. We have a camera above the animal so we can get the XY coordinates of the animal. And the blue dots, one blue dot, that is this pop, 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 action potential. And you remember the place cell maybe, that was just one such field, but this cell had many fields. And what was so special is that these many fields are so regular spaced that you can fit in equilateral triangles. This is biology, it's not math or computer science or whatever. And still, these are so precise. So we decided that these grid cells and many cells would show those patterns. They could function as a universal coordinate system or the metric of the brain. But we started with this question, what is the relationship between what is happening in the entorhinal cortex and the hippocampus? And what we also saw is that if you go more ventral, more down to earth in the brain, then the resolution of these grid cells will change. So you have fields that are small, the more dorsal you are, and then they increase when you go more ventral. And models, just after we discovered the grid cells, suggested that if you align grid cells of different sizes on top of each other, as you see here, the small ones and the bigger, then you add up the activity here in the middle, but you cancel out the activity in the edges. And what do you get? You get exactly one field as we saw in the hippocampus. So at least we think that some of these grid cells could provide such a place field in the hippocampus, but we also know that other cells talk to cells in the hippocampus. So another question that we asked is, how can a biological system be so accurate, placing these fields so accurate in the environment over and over again? So if you come back the next day, it looks exactly like this pattern. So this cell needs information about velocity, the direction of the animal, and also the heading. Could we find such cells in the brain? And we already heard about Jim Rank and Taube. They found such cells in the dorsal presubiculum. Here we asked, do such cells exist also in the entorhinal cortex? And yes, again, with fantastic postdocs and PhD students working in our group, we found then head direction cells upstream of the grid cells. And again, now you might recognize how these head direction cells fire. Another question we had was about the speed, because velocity, as I said, is both direction and speed. And can you imagine, so these are old data, but can you imagine how difficult it is to understand whether there is a speed signal in the brain? How can you do that? Because you have to control the speed of a freely moving animal. And the only way we could do that, to have perfect control, was here with another wonderful postdoc at the time. We constructed a path and a car, and the animal had to run like a Flintstone in order to keep awake and to keep the theta activity, for those who knows that. And then we could control the speed of the car with the computer. What did we see? When the animal was driving north in a high speed, the cell would be firing a lot. And when it slowed down the speed, the firing rate dropped. And when it was driving the opposite from north to south, you would see the same pattern. So these cells typically respond to the speed of the animal. But as you know, animals don't drive normally. So if you just let them run spontaneously in the open field, we could still find many of such cells. In this case, 358 neurons had this relationship between the firing rate and the speed. So these are different cells that we recorded in the open field. So there's a linear speed firing rate relationship. And giving this velocity input then to the grid cell has a precise firing in the environment. So 50 years of recordings in the hippocampus and in the entorhinal cortex, where we found all these different functional cells. I didn't even tell you about the border cells, but they're easy. They're just telling you about the boundaries in the environment, and then boop, 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 and then going away, boop, 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 going away. So, and then the other types of functional cells, like object vector cells, these cells would then tell you the distance to objects and also the direction. So we could stop the story here, but of course we don't, because these are the single cell responses. And as I said, in the human brain, there are more than 80 billion neurons. So in order to understand the algorithms of the brain, we need to understand the population and how the population is working together. And then I should almost ask Eva to come and switch with me, because now we have to start to think big data, computer science, mathematics, and so on, in order to understand maybe the chaos that all these electrical signals in our brain are doing. Luckily, we got another Nobel Prize last year, John Hopfield, and he and others suggested attractor networks. So this is a way to try to, so of course, he didn't work in neuroscience, but his models would be very useful for us working in neuroscience, because he and others suggested then how recurrent networks that the cells talk to each other, how certain activity states could emerge. One way to think about this is that he was talking about point attractor networks, and these networks, they are not anatomical networks. It's how the cells talk to each other, how they are connected. So you can't see this in real life. But the point is that you have this activity that can converge to different states. So if I smell some really good wine, like I did with, at dinner yesterday, I might be sent back to Villeneuve, and I might remember the beautiful party we had. And then my memory goes then downhill, and it's stuck here until I get a new signal and I'm sent to another winery in Alsace, for example. So what we know is that activity here at the surface, think about the moon landscape, is very unstable, but it's very stable in the craters. There are other attractor networks that are important to understand space. So here you don't have point attractor networks, but you have continuous attractor networks, no craters. So any place in this network is equally probable. And this is called a continuous attractor network. And it's very useful when we want to explain head direction cells and grid cells, for example. So I'm going to walk you through, for example, the head direction system with the hat on as a continuous attractor network. I told you about the head direction system, and this is a bump. Again, this is not anatomical, this is not about space, but it's about cells that respond to a certain direction. And then there's all these cells here that are warm colors, they would then fire whenever the animal is pointing its head in one direction. These cells have asymmetric connections to other cells, and the others here will be silent. And the whole point here is that you can't jump, you can't cheat. You have to follow poop, 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 or poop, 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 poop. And that is what I want to see in this, show you in this video with the cat. So these attractor networks, they force the activity within a manifold. And this manifold for the head direction system, that is a ring. So you can't get the ball to jump from north to south. And it's the same with the states of the cells. And the cat can play forever. So you see that the only thing that can get the ball to move is that you get some angular speed from the cat or from where the head is pointing. Now it becomes a tiny bit more difficult because now it's not only one dimension, but we have to go to 2 dimensions. And now we talk about models for place cells. I'm not going to spend so much time on this because it was a very good model, but it had some problems. What I can say is that if you think about, again, place cells being active in one spot, and then other place cells being close to these cells also active, inhibiting the cells that are not active at this place. And then when the animal is moving around in this space, then you get this bump of activity. Some people would then call it, this is the Mexican hat, excitation inhibition model with this continuous attractor models. So the activity is then translated throughout this. There's one problem with this model, and that is that, like the fairy tales saying that when the earth was flat, then there was something, a troll or something terrible at the boundaries of earth. Because you have to go across, and you couldn't go around like we believe now. When it comes to these continuous attractors, it makes life so much easier if you can connect it and make the map, this attractor map, like a torus. So this was suggested early on and was in fact a problem when they tried to explain place sets because you got a grid pattern. And that was a good luck for us because we needed models for the grid cells. So if we jump then to the grid cells, you can now understand how you can go from this 2D surface to a torus. Because you can go, so these are 3 different grid cells. You go to rhombus, you connect the different sides of the rhombus, and then you get the donut. So you can play with all these things the way you want. But we don't only play in science, even though we are kind of children, because we love to play. We also like evidence. We like to prove things. And in order to prove such theories, we need not only the theories, but we need new tools. And what type of tools could we get in order to address such questions? Do we have a donut in our brain? Luckily, Edward and I in our lab, through, again, fantastic postdocs in our lab, we were allowed to join a consortium where people developed, then run from Janelia Farm and then made in Belgium, they made microchips where you didn't only have these small sensors having only being able to record a few cells at a time, but here we could record thousands of cells because it's a microchip and so many channels. So this device is called the NeuroPixels 2.0. It was published in 2021 in Science if you are interested. So what can we do with this? We can record thousands of cells. And in this example that I have here, we recorded 149 grid cells of a certain size. When grid cells come in a certain size, we call it this sized grid cells that they belong to a module. So these cells belong to module 1. If we want to understand the state space, how these cells are coactive, we have to look into 149 dimensions of activity. If you see this here, this is the raster plots. So that means this is cell 1, cell 2, cell 3, here, the activity. And here we can look at the population activity for certain times. And since cells are not pacemakers, typically, this activity, when you move this slice in time, will vary a lot. And then to understand this variation, we need to do something magical, and that is to reduce all this variability to fewer dimensions. Luckily, there are mathematical tools to do that, and here you see an example of such a visualization tool called UMAP that can reduce all these dimensions down to 3 dimensions because the cells are also correlated because these are grid cells. And do you agree that this could be a donut? Do you see the circle with a tiny hole? So you could turn it around and you could agree. Again, this is just a visualization. It's not a quantification. So it's nice to see. We celebrated in the lab when we saw it because it would fit to the models telling us that there should be a torus in the brain, the grid cells. But we would like to know how this is in a more quantified way. And again, we had to collaborate with fantastic mathematicians at our local university. You find the references here. So it was Niels Boas and Ben Dunn and Erik Hermansen and these people. So they could use a method to check the activity, the state space of the activity of these cells, and then decide whether this state space follows a torus. And how could they do that? When they use something that is called persistence homology, they can, all these dots, that is these red bars with the activity, and they can fit a circle around each dot and then find out when the system is fully colored. And that is exactly when you get one bar in this dimension, and then you do this, repeat this for different dimensions, and there are these recipes saying that if you have 1 in 0 dimension, it's 1 manifold, it's 1 structure. Then you have 2 in dimension 1, that is these 2 red circles here, the 2 axes of the torus. and then there's one hole in the middle. So you can compare the Betty numbers here with the Betty numbers of a circle and a sphere. And here you have these different dimensions, and the orange, that is the shuffled, and the green bar, that is the significant. So if you can read out these bars, you know what kind of manifold the state space of these cells would contribute to. So we asked then, together with these groups, do we have a torus if we check grid cells of one module? And if you remember from the last slide, I think you could be convinced that this is a clear torus. What was so strange was that when the animal was walking, not in an open field, but in a wagon wheel environment where you don't see grid cells that clearly, still the torus exists. What was even more surprising, and this is in fact Edward's and my grandson, Alfred, sleeping. When we are sleeping, I think this happens in our brains, and when the animals are sleeping, these grid cells, they follow this torus activity. And they can't jump out, like the cat couldn't move the ball out of this frame. Our activity of the grid cells have to follow the activity of the torus when we sleep. And in order to know if this is correct or not, you can use this information and then go back to the environment or the rhombus and decode the activity from the torus. And what you see here is that cell 1 has fields in the rhombus that corresponds both in the open field in the two different sleep stages. And the same for cell 2, cell 3, cell 4, cell 5. So we are really happy about this result. It means that, the torus exists. And this is just to sum it up. I have a feeling that I don't have that much time because I have more to tell. But what is important here is that this torus, this grid map is internally generated. It doesn't need any sensoric input to just go around and round and round. Why is this interesting? I will tell you in a bit. Just enjoy the ride in the torus. Because now we have the knowledge and the tools, and I have to say before I talk about this, this is not published, and I know that you are going to publish my talk. So Edward and I discussed back and forth, can we tell about this story or not? And then we thought this is such a nice story that we take the chance to tell it. So I hope you like it. The question here is that, is it so that the grid cell circuit in the entorhinal cortex, is it inherited or is it through experience? And of course, we heard about ancient times, and the philosophers have always wondered how brain functions come about. And you have the empirism, for example, with the tabula rasa, that you have the blank sheet, there's nothing there when you are born, and then everything that you become is just because of your experience. This is very different from the Immanuel Kant's version, because he said in order to experience, you have to have some faculties of mind. And he suggested that we are born with ways to understand space, time, causality before we start to experience. And of course, Edward and I were both psychologists, and these are questions that we struggled with all the time when we were students. And now, as scientists, we finally are allowed to start to ask such questions ourselves. I just have to mention first about the development of the rat. The rat is born at P0. The first 2 days alive, then olfaction and whisking start to work. Only 12 to 14 days after they are born, then their eyes are open and the ear canals. What I learned from my children is that this happens in the humans when it's still an embryo in week 18. At P15, when they are 15 days old, then the animals start to leave their mom and the nest and their siblings, and they start to explore. So at this time, if Kant was right, the animals, they would need a device that is there already before they start to experience the environment. Do you agree with that? And then in Norway, I just learned that we wean the animals earlier than many other places, but we wean the animals at P21. Okay, so earlier, we and others, the London groups, we asked this question: when do we see grid cells? That was before we had the tools to study the torus. So we asked, when can we see grid cells in the baby rats? And both groups, we decided that it looks more normal at around 3 weeks after birth. So that means that they have been walking around then without having perfect grid cells for a while. So this would be a tiny issue maybe for Kant. We also did an experiment where we asked, is the experience important for grid cells to develop? And the way we did that was to raise animals in different environments. And you see the environments here. They were opaque walls in the two of the environments, the sphere and the cube. And then the rest of the animals, they lived like our animals live normally in Norway, with a lot of toys, with a lot of friends, and they could, of course, see whatever they wanted. So what happened when animals were raised like this? If you see first how the sphere animals' activity of the cells looked like, it was very, very difficult to see any grid cells at all. These pass the criterion, but they're not perfect grids at all. In the box, you see cells that are much more similar to grid cells, and of course, in the enriched environment, there are a lot of grid cells. And here we sum it up. So you see the percentage of grid cells high in the two different environments, but not in the sphere. So it seems like the animals need experience with straight walls and corners in order to develop normal grid cell activity. Can this be repaired? Yes. So when we trained the animals in a normal open field, it took around 1 week before these animals started to develop normal grid cell activity. So now we know a tiny bit about when the grid cells appear. What about the torus? Maybe Kant was thinking about the torus when he suggested that space is there before we experience space. how can we study this? Again, if we remember what happened with animals, we decided to study these baby animals with the new tools, the neuropixels, because one neuropixel probe is only 1 gram. So they're not affected by these sensors at all. And then we wanted to implant such animals way before they started to leave the nest to ask whether there is a torus. And of course, before they leave the nest, they're just sleeping. But we know from before that the torus is going around and around and around even when we sleep. So we should be able to study these animals and ask the question whether there is a torus in sleeping, non-exploring animals. Here we couldn't go in and say we want to check the grid cells because they didn't move in the environment, but we know that grid cells are correlated, and that meant that we could use these matrices, the correlation matrices, and then check the high correlated activity here and check whether these would, the state space of these cells would follow the pattern of a torus. And with the UMAP visualization, you see that happened, both at P10, P11, that is 5 days before they start to explore the environment. And when you see the barcodes, not as perfect as in the adults, but still indicating that there are tori in toroidal manifolds even at day 10 after these animals are born. We can decode the activity in the rhombus, and you see that the decoding looks gorgeous. What happened then? What magic happens when you see that at P9 there's no torus, at P10 there is a torus? When the animal is a tiny baby, you have these twitches, you have other types of input, and that is what you see that these animals respond to. So here again, you have a raster plot. This is at 8 days old animals or 9 days old babies. And you see this stripe of activity where the cells are active and where they are silent. There's very little crosstalk between cells. But suddenly, at P10, when we see the torus, the network is changing. And in honor for the Hungarian Neuroscience Society, you see that inhibitory connections are one of the important changes in these networks in order to generate the torus. So we can then conclude, should we give Immanuel Kant a hooray? He deserves that. Hooray! And what we hopefully have learned today is that these networks are develop intrinsic activity and connectivity by P10, and then you see the torus, and so on. So I don't want to say too much about this, but then I think the last thing that I'm saying here is, okay, I have a few slides more, sorry. Because what is also important, I'm not going to show slides on that, I'm just going to say it, that now we have the tools and the concepts to uncover these algorithms. We can also then start to hopefully understand what happens in the diseased brain. What I can tell you very, very briefly is that in patients with Alzheimer's disease, It starts in layer 2 of entorhinal cortex, and the cells that I presented, the grid cells, the object cells, the border cells, the head directions, no, not the head directions, but these other cells, they are the first ones to die when people get Alzheimer's disease. We know that the symptoms of Alzheimer's disease is that people don't remember, they can't find their way, and they have trouble with time. So, I don't know if you recognize this guy. He was the Secretary General of NATO. He was our former Prime Minister. He is now our Finance Minister, Jens Stoltenberg. And he said, knowledge trumps everything. It is each other's work and each other's knowledge that we will live on in the future, which is the vision of the academy in Hungary and all other places where we do science. And then this is, hopefully, if we can get the sound. Yes. And I told Katalin Carico, this is especially for you because you have shown so much persistence. And all of us, we have a PhD, and I think we all know how it is to struggle in science. Take a break, think about it.
Wonderful. Thank you so much. We really appreciate
Thank you.
it. It's an amazing story.
Thank you.